cellular ultrastructure. So that basically means what we find inside our cells. So if we first of all draw an overview, label the parts, and then we need to look at what all of those parts do, what all those organelles do. Okay, so first of all, we're going to be looking at what we call a eukaryotic cell. Okay, so there's two types of cell, eukaryotic, and the other type is prokaryotic. And we need to know the definitions of these first of all. So if something is eukaryotic, it means that it's an animal or a plant cell. If something's prokaryotic, it means it's a bacterial cell. And why do we group things in these two groups is determined by whether or not they have membrane-bound organelles. So eukaryotic cells do have what we call membrane, you need to know this exact phrase, membrane-bound organelles. Okay. And the difference between eukaryotes and prokaryotes is the prokaryotes do not have membrane-bound organelles. So let's have a think about what a membrane-bound organelle is. So we know some organelles that we find in cells. So let's have a think about whether they're membrane-bound or not. So let's have a think of a nucleus. So here's our nucleus. We've got the DNA inside. Has it got a membrane around it? Yes, it has got a double nuclear membrane around the outside. So that would only be found in eukaryotes. Let's have a think about a mitochondria. So mitochondria, site of aerobic respiration. Has it got a membrane around it? Is it membrane bound? Well, it's got a single membrane around the outside and a folded or smooth membrane, I should say, around the outside, folded membrane in the middle. So yes, membrane bound. So it's only going to be found in eukaryotes. What about a vesicle or a lysosome? Yes, those are membranes around the outside of them. Definitely membrane bound, only found in eukaryotes. What about endoplasmic reticulum? Well, these structures here are called membrane-bound sacs. So those are definitely going to be membrane-bound, definitely only in eukaryotes. What about the Golgi apparatus? These are definitely surrounded by membranes. Each of these little vesicles coming off, membrane-bound. Golgi sacs, membrane-bound, only going to be found in eukaryotes. However, these little tiny dots, which we call ribosomes, they do not have membranes around them. So they are found in eukaryotes and in prokaryotes. Okay, you would find those in both. A prokaryote would also have the membrane, the cell surface membrane, the plasma membrane around the outside of it, but it wouldn't have any membranes around the organelles inside it. So all of these organelles are only found in eukaryotes. Now, because this course is a human biology course and humans have eukaryotic cells, it is going to be the eukaryotic cells that we're really interested in. So first of all, it's unlikely that you'll have to draw one. Probably you'll either see a micrograph picture or a diagram and you'll have to label the different parts. So first of all, let's start off with this layer around the outside here. So this layer around the outside is what we would have called a cell membrane. Now be careful with your language because it's obviously a level 3 course. We need to up our language. So this can be called a cell surface membrane, or you can call it a plasma membrane. And we do obviously need to know the structure of this membrane with the phospholipid bilayer, etc. And we need to know the function of it as well. So if we have a look at the structure of our cell surface membrane, oops, doesn't change today. Structure of our cell surface membrane is a phospholipid bilayer. We go into that in a lot of detail, don't we? Phospholipid bilayer with the fluid mosaic model. make sure you know what those words mean. It's got proteins embedded in that layer. So, and proteins. And the job of that, the function of it, is that it controls what can go in and out of cell. Can move in and out of cell. So, because it can let some things through but not other things, we say that it's semi-permeable. However, semi is a bit of a kind of GCSE word, so we're more likely to say partially or selectively. So partially or selectively permeable. Okay, so some things are going to go into the cell, some things are going to move out of the cell, some things go straight through the phospholipids, some things have to go through the protein, but it controls what can go in and out of the cell. And it separates this substance inside the cell, which is called the cytoplasm, from the substance on the outside of the cell, okay, from this outside environment here. So it's just a membrane that separates those two places, really. Okay, so controls what passes through and separates the inside of the cell from the outside of the cell. So the next part of our cell, draw our cell here, we've learnt about the membrane around the outside. The next thing is the nucleus. So GCSE version, we would have called this the nucleus. Okay, we're now at level 3, so we need to add a little bit more detail about our nucleus. So we know that the nucleus contains the genetic information. So the genetic information is the DNA. Okay, DNA folded up into chromosomes, isn't it? And that genetic information basically controls everything the cell does, because the genetic information codes for proteins, and those proteins control all the reactions. An example of a protein is an enzyme, and enzymes are what catalyse reactions. So, in a roundabout way, we can say that it controls the cell's reactions. Now, another way of saying that it controls reactions, like all the reactions that happen, are called the metabolism. So, we could say it controls the cell's metabolism. Now, in the nucleus, we're always going to see a little darker region within there. And that little darker region in the nucleus, just there, is called the nucleolus. Now, the nucleolus is very dense. It's spherical, like a ball, like a circle, 3D ball. It's a sphere. And the job of it is that it makes RNA and ribosomes. Fancy word for makes is it synthesizes RNA and ribosomes. Okay. Now, around the outside of our nucleus, we've actually got, I change that colour to white. Around the outside of the nucleus, there's actually little holes called nuclear pores. Okay, so the nucleus has little tiny holes in the membrane surrounding it. So the membrane around the nucleus is called the nuclear envelope. Okay, and within the nuclear envelope, we have nuclear pores, which are basically like little tiny holes. And that allows the movement of molecules into and out of the nucleus. Okay, so allows movement in and out of the nucleus. And a very famous example of something is that the mRNA that gets made in the nucleus comes out of the nucleus and goes to the ribosomes in order to make proteins. Now, a little bit more about our DNA. So our DNA is folded up into chromosomes, and we talk about it containing chromatin. So chromatin is basically oops. Where's it gone? It's basically like strands of DNA that then fold up into the proteins. Oh, there we go. Where's my spelling corrector? I just have to rub it out. There it is. So remember, sorry, here we go. Something was leaning on the mouse pad, that was why. Chromatin. So remember that if you want more detail, you can talk about how that we've got the chromatin, kind of like all little strands of DNA, and that folds up with proteins into chromosomes. Okay. So we could just talk about the DNA or the genetic information, or you could go into much more detail. Okay. Let's move on to the next organelle. So, I think first of all, before we move on, let's first of all do a bit of a recap. Just make sure we can remember it as we go. So around the outside of the cell, we've got the cell membrane, which we're going to call either the cell surface membrane or the cell plasma membrane. Remember that that membrane is made of a phospholipid bilayer with proteins embedded in it. Remember that the job of that is that it's selectively permeable or partially permeable, so it controls what enters and leaves the cell. Then remember that we've got the nucleus that's got a nuclear envelope around the outside with these little nuclear pores to control what can go in and out. That we've got the genetic material made of chromatin and wrapped around proteins to fold up into chromosomes, which controls the cell's metabolism because it codes for proteins. Then we've got this darker, dense spherical region called the nucleolus, which synthesizes or makes the RNA and the ribosomes. Okay. So next up, we've got something called the rough endoplasmic reticulum. Now, this can be an extension of the nuclear envelope. So we draw it like this. It's not individual sacs like the Golgi, it's all interconnected. Okay, that nuclear envelope is actually a double envelope, like so. And you can see how the outside part of that envelope connects with the rough ER. Now, the thing that makes this ER rough is that it's got these little spots all over it, and those little structures are the ribosomes. So we talked about how RNA and ribosomes are made by the nucleolus in the nucleus, okay, and these are these little ribosomes here. Now, sometimes ribosomes are found loose free in the cytoplasm as well, but there are lots of them found on this structure. When you do arrows, make sure they do touch. You should draw the line of your arrow with a ruler as well. Sorry, it's quicker for me to not, but you should do. Which is called the rough ER, which stands for rough endoplasmic reticulum. Okay, so a little bit about the rough endoplasmic reticulum, or the RER, as you might want to summarize it too, is that it synthesizes and transports proteins. Okay, so in very simple terms, if you need a a description of the function. What it does is it synthesizes, which means makes, and transports proteins. So let's look at how it does that. So if we were to zoom in on the endoplasmic reticulum, it might look something like this. Okay, so on the surface, we've got the ribosomes. Now, a ribosome actually has one small subunit and one large subunit. Now, a fancy way to describe the size or the density of ribosomes in eukaryotes is we say they're 80S. Prokaryotes are 70S, eukaryotes are 80S. So what our ribosome here will do is it will make the protein, synthesizes protein synthesis. Now, once the protein is made, what will happen next is that protein will go into the rough ER and it'll be transported through this network of these flattened sacs, membrane-bound sacs, and then eventually the protein will need to come out of the rough ER, and the next place it goes to is the Golgi. And how it gets to the Golgi is it gets put into a vesicle. Okay, so it goes into a vesicle, and that vesicle will continue on its journey through the cell. So when we say it synthesizes and transports proteins, that's what we mean. It synthesizes the protein on the ribosome there, and then the green is showing us how the rough ER, is transporting protein. Okay, so it's doing those two roles. Now, if you were to describe this shape here, the way you need to describe it is that it's a network of membrane-bound flattened sacs. Learn that description. So it's a network of membrane-bound, because there's membranes going all the way around. Membrane-bound, flattened sacs. Okay. Now, we say that it has cisternae. What that basically means is the way it goes out and in, out and in, out and in. So we call that shape cisternae. Okay, so it's got cisternae, and the cisternae are then covered in ribosomes, and the ribosomes is where we're going to make the proteins. Okay. So where do our proteins go next? We've said that they go on to the Golgi. So within our cell, so remember the different parts. So we've got the cell surface membrane or the plasma membrane. We've got the nucleus, so with the nuclear envelope around the edge with the nuclear pores to control what can go in and out of the nucleus. We've got a darker, denser region here called the nucleolus that makes and synthesizes RNA, makes or synthesizes, that means the same thing, RNA and ribosomes. We've got the genetic material in the rest of the nucleus. Then we've got our rough ER, which is rough because it's covered in the ribosomes that are doing protein synthesis. And our protein then gets put into a little vesicle, which is transported to the Golgi. Now, the Golgi in some ways looks similar to the ER, but in other ways it's different. Now, these little things that I'm drawing, they go round in a sort of almost like a rainbow shape, although this one looks like it's upside down. And they should be thinner in the middle, more bulbous, fatter, sticking out at the ends. Can you see I'm trying to make them a bit wider at the ends? A bit thinner through the middle. It's tricky drawing on here. And we call that then the Golgi apparatus. Okay, so lines must touch. Draw the line with a ruler. This is the Golgi apparatus. You can just call it the Golgi, but the full name is the Golgi apparatus. So this is where our protein arrives after it's been made at the rough ER, transported through the rough ER, transported then in a vesicle. We never labelled those, did we? These little transport things are called vesicles. It's just a little ball with membrane around the outside carrying the protein in the middle. And it comes along to the Golgi apparatus. So at the Golgi apparatus, this is where our newly made proteins arrive, and the kind of description of the function is that they modify and package the proteins. So let's have a look at what modify actually means. So modify often means to add a little carbohydrate onto the protein. So e.g., add carbohydrate onto the protein. Okay. And when it says package, what it means by that is just the same as when it came from the raffia. Once it's gone through and been processed, it again gets put into another vesicle that can then transport the protein once it's been finished to wherever it needs to go. So this is another vesicle. Okay, so packaging, it just means put it into a vesicle so that it can be taken wherever it's needed. That might be somewhere else in the cell. If it's an intracellular protein, that means inside the cell. Or it might be that it needs to leave the cell. It might be an extracellular protein. It might need to go out of the cell. Okay. Lysosomes can also be made there. So if the protein is a hydrolytic enzyme, then when it's put into a vesicle, that vesicle is then called a lysosome. Okay, so let me explain that again. Enzyme. So hydrolytic enzymes are basically digestive enzymes that break down like old organelles or old cells or old things that we don't need, even pathogens that we don't need. We want to break those down, don't we? Pathogens are like disease-causing microorganisms. So a vesicle is just a little circle of membrane with some kind of molecule inside it, and it's just transporting it around. Okay, a lysosome is basically just a version of a vesicle that contains hydrolytic enzymes. So if the protein that was made by the endoplasmic reticulum that was then transported to the Golgi, that was modified in the Golgi and packaged off into a vesicle, if that protein was a hydrolytic enzyme, then that vesicle is now a lysosome. So we can say that the Golgi make vesicles and also make lysosomes as well. Okay, let's move on. So recap where we're up to so far. So we've got the cell surface membrane or plasma membrane, controls what enters and leaves the cell because it's got a partially permeable membrane. We've got the nucleus, which has got a nuclear envelope with nuclear pores to control what goes in and out. The nucleus contains the genetic material. We can talk about chromatin folding with proteins to form chromosomes. It's got a little denser, dark region that we can identify as the nucleolus, which makes ribosomes, makes RNA. Then we've got the rough ER, and the rough ER has the ribosomes on the surface, and it's going to make proteins, protein synthesis. Those newly made proteins go in vesicles and move on to the Golgi apparatus, where they get modified and packaged off into more vesicles. Those vesicles might then leave the cell or they might stay in the cell. Okay, so that's the journey of basically a protein. The rest of the organelles are a little bit different because they're not in that protein journey. So the next one we're going to look at is the mitochondria. So it's got a double membrane. The outer membrane is smooth and the inner membrane is folded into this shape like this. So mitochondria are the site of aerobic respiration. Single would be mitochondrion, plural mitochondria, and it's the site of aerobic, just write it above, aerobic respiration. Remember that respiration releases energy. Don't ever say it makes energy. Energy can't be created or destroyed. So it's the site of aerobic respiration which releases the energy from molecules like glucose. You need to be able to talk about this structure. You need to talk about how it's got a smooth outer membrane and it's got a folded inner membrane. So it's a double membrane, smooth outer membrane, folded inner membrane. And that inner membrane, where it folds into this sort of in and out, in and out, in and out structure, we need to know that that's called the cristae. So if I was to draw it a little larger, okay, smooth outer membrane, folded inner membrane, and this folded membrane here, that's the cristae. The middle part in here, in the middle part there, it's almost like it's got a liquid inside, like equivalent to the cytoplasm of the cell, I suppose. It's got this middle part, this central part, and that's called the matrix. And you need to be able to label that. You need to be able to label that this folded membrane here is the cristae, and this middle kind of empty space in the middle, what looks empty in a diagram, is called the matrix. Now, if you imagine that this is the shape of a sausage, if you were to cut a sausage sideways, long ways, longitudinal, the shape of the sausage would be like this. However, if you were to cut your sausage the other way, transverse, a cross section of it would look like that. So you need to be happy to identify mitochondria in their classic way of being like this. Also, that they might be cut in this transverse way, in which case they look like this. The other thing is, if you're trying to recognize mitochondria from electron micrograph images, the cristae is more like this, with even thinner. Okay, so just be careful that you're not looking for the stylized version. It's like loads and loads of really thin, really tightly folded. I'm bored of drawing it. You see why we never draw it like this? It takes too long. Okay, but you're looking for something more like that. And in fact, often it might look to your eyes more like that. Okay, you can't always see this clear wavy line like a wave. Okay, they're actually really, really thin little folds. So the mitochondria is the site of respiration, of aerobic respiration. Now, in fact, it's not all of aerobic respiration that happens in there. It's actually just the later stages of respiration. But we tend to say aerobic respiration occurs in the mitochondria. But if you wanted to be picky about your detail, it's actually the later stages of respiration that happens in there. Okay, so that's our mitochondria, site of respiration. Make sure you can recognize the shape. What else have we got? Oh, I know, the smooth endoplasmic reticulum. Now, this is probably the one that people get wrong the most when they're labeling cells. Well, not necessarily labeling cells, but when they're talking about the function of organelles. So if that's our nucleus. We've got rough ER, which we can recognise because it's got these ribosomes on. Golgi, which we can recognise. It's more like this sort of rainbow shape. Should be more bulbous at the end, so I'm not very good at drawing it. Vesicles, obviously, just like circles. And then we've got some more endoplasmic reticulum with these joined sacs. Remember, they're not separated like Golgi are. They're all joined together, but there's no ribosomes. This is called the smooth ER. And it has nothing, it has nothing to do with proteins. Everyone, I think, gets so bogged down with the ribosomes making proteins, transporting proteins, Golgi modifying proteins, packaging proteins. You've heard proteins so much that we go into talking about proteins when we come into smooth ER. And smooth ER is not doing anything with proteins. Okay, not proteins. So what does it actually do? Well, what it does is it makes carbohydrates and lipids. So rather than saying makes, try to say synthesizes. So it synthesizes carbohydrates lipids. So what are the three biological molecules groups that we learn? Carbohydrates, lipids, and proteins. It's only proteins that are rough ER, and it's carbohydrates and lipids which are made in the smooth ER. Okay, so don't get those muddled up. It's really important that we know that there are no ribosomes. Okay, hence they cannot make proteins. So the lipids and the carbohydrates are made in the smooth ER, and we can also say stored. So synthesizes and stores carbohydrates and lipids. There's no ribosomes, so it can't make proteins. It has nothing to do with proteins. It just makes carbohydrates and lipids and stores the carbohydrates and lipids. So that one's a little bit different. The next thing we've got to mention, draw a cell surface membrane, plasma membrane, nucleus, nucleolus, rough ER with the ribosomes. Hopefully I'm repeating myself enough that we're starting to really quite easily remember it, hopefully. Golgi apparatus. So rough ER makes the proteins, transports the proteins through, puts them into vesicles. Golgi apparatus modifies the proteins, packages them into more vesicles, off they go on their way. Smooth endoplasmic reticulum, nothing to do with proteins, makes and stores lipids and carbohydrates. So the next thing we've got is centrioles. Now, centrioles will usually be drawn in a sort of pair, with one like that. They're usually drawn crossed over, and one like that. Sometimes pictures are drawn much longer and thinner, so you might have one like that. One, oops, not a good drawing. One something like that. And these are centrioles. Now, the centrioles are involved in cell division when a cell divides. Okay, when one cell divides into two cells. And you remember something about mitosis, when the cell divides by mitosis, is what happens if this is our cell, then our chromosomes line up in the middle on what we call the equator of the cell. And then what happens is the centrioles, which is what we're learning about now, are at the poles of the cell. So we got a centriole here, centriole here. They're the poles of the cell. Arrows should touch and be drawn with a ruler. Sorry, I'm not setting a good example. And those centrioles send out something called spindle fibers. So the spindle fibers reach out to those chromosomes on the equator, like spiders' webs, and they get hold of these chromosomes. You can see that each chromosome has been split in half, one chromatid going to each pole. Okay, and then the centrioles are going to shorten. Sorry, the spindle fibers are going to shorten. So here's our centrioles, here and here, and those spindle fibers shorten. So if they're then only that long, then they're pulling the chromosomes towards the poles. And eventually what will happen is the cell membrane will split down the middle, down here eventually, and it will form two new cells. So in this context of this, what we need to know is that the centrioles make the spindle fibers attach to the chromosomes and pull them to the poles in cell division. Okay, that's their job. So let's add on that they, in cell division, they make spindle fibers. So you need to be able to recognise pictures of them. and you need to be able to describe what it is that they do. Now, sometimes you might hear the word centrosomes. Now, this definition of centrosomes is that they're made of two small microtubules called centrioles. So this structure here with the two, that you could call centrosome. Okay, and it's made of two microtubules called centrioles. And then we talk about how one centriole goes to each pole of the cell and produces these spindle fibers. The spindle fibers then attach to the chromosomes. The spindle fibers shorten, pulling the chromosomes to the pole, ready for the cell to split in two to make two cells. And I think that probably brings us to the end of it. So make sure that you can draw that, not necessarily draw really, but just make sure you can label a cell. Make sure you can do it from a diagram, and make sure you can do it from an electron micrograph. And make sure you know the function of each one. So sometimes you might need to describe the structure of the organelles, and sometimes you might have to describe the function of the organelles. So just make sure that you can do both of those things. Okay, and that's the end.